Hello, I'm Clare Stanford, and I'd like to introduce my colleague David Heal, who's going to be talking later, and we're both going to be covering the use of preclinical models in psychiatric drug discovery and flagging some important points that we think are going to crop up later in the discussions today. So I'm going to be dealing with mode of action and efficacy, and David is going to be covering tolerability and safety, both of which bear on benefit risks and public and society and so forth. So the first thing really to consider is what would be needed for an abnormal animal behavior to qualify as a model of a human psychiatric disorder. Well, I'm going to be using depression as a good example of that. And there are various ways of diagnosing depression, various systems. Here we see that they have a lot of features in common, but there are differences between the two. But they list several different features of the disorder. And for example, DSM-5 requires human patients to express at least 5 of 9 symptoms for at least 2 weeks in order for the patient to qualify for a diagnosis of depression. So depression isn't a single disorder, it's a collection of problems. And these problems can differ from one patient to another. They have different weightings, and each of them can wax and wane over a period of time. So we can have 2 depressed patients with completely different aspects of depression, but both of whom are diagnosed with that problem. So how are animal models doing as a model of depression? Well, I've reproduced those symptoms and signs really on the left-hand side. And in the purple, those are the features which arguably could be evaluated in rodents. Anhedonia, for instance, is thought to be the rodent equivalent of a loss of sucrose preference when animals have experienced a bout of chronic mild stressors. Rodents, of course, have a very sweet tooth. They die for Ribena or caramel-flavored carnation milk, but they lose that when they've had a bout of stressors. Low self-esteem is arguably measured in the resident intruder test, where the intruder develops a submissive behavior eventually. Cognitive deficits, ironically, are quite straightforward. I mean, it requires expertise, but it is possible to measure when a rodent isn't concentrating, or when it's lacking in accuracy, or when it's slowed down its learning behavior. It's also quite straightforward to measure changes in weight, appetite disturbance, self-neglect, and changes in sleep rhythms, for instance. But there are key features of depression which we will never be able to measure. They include things like guilt, feelings of hopelessness, suicidality, and hypochondria. So in looking at the literature, it's, in my experience, it's never been approached as a model of depression that has measured many different features of this type in a way that would be analogous to a diagnosis of depression in humans. Most tests at best simply measure motor behavior, and most studies, even if they're conscientious, only measure 2 or 3 of these aspects, these aspects of depression. So recognizing that, many researchers don't call it a model of depression, they call it a model of depression-like behavior. But it's never quite clear what symptom or sign of depression is actually being modeled. The literature is really rather vague on that. Important question is each of these behaviors an endophenotype? In other words, does the abnormal behavior map onto a neuronal network or even a genetic mutation? Should we really be looking at illnesses like depression as a collection of endophenotypes rather than as a unitary disorder? And that would reflect the situation, for instance, like latent inhibition in schizophrenia, where there's a lot of evidence that latent inhibition does map onto a genetic mutation, although that is actually controversial. There may be subtypes of latent inhibition. Well, even if we regard them as endophenotypes or individual aspects of a problem in depression, there are still complicating factors. For instance, abnormal behaviors are not confined to a single disorder. For instance, social withdrawal, cognitive deficits are evident in Alzheimer's disease, schizophrenia, and major depressive disorders, as Hugh Marston would describe. And also, the disorders can coexist. For instance, it's quite common for depression to coexist with ADHD and for depression and panic disorder to appear in the same patients, for instance. So we need to be very careful to validate what aspect of the disorder we're actually measuring and what disorder is being modeled. So how are we doing? These are the procedures which are used to measure depression-like behavior in rodents. And there's a long list. This isn't an exclusive list, but very early studies, for instance, use rezapine-induced hypothermia and hypoactivity, and it is still used in some countries in the world, even though hypothermia is not ever actually seen in depression. Learned helplessness is another good example, where animals experience an escape deficit after a period of inescapable stress. But even the people who developed it in the 1970s are not at all clear about what learned helplessness actually is as a human equivalent. It has been described as PTSD, and nowadays it's thought to be an element of passive coping. The olfactory bulbectomy model is probably the best or closest to a model of depression, but even the people who developed it recognize its limitations. But it does have many features in common: endocrine aspects, behavioral aspects, and importantly, the abnormalities are only reversed after prolonged treatment with antidepressants. The tail suspension test, where mice are suspended upside down by their tails, is often used as a model. But if you think about it, if two human beings were suspended upside down against their will, it's very doubtful if a difference in their struggling behavior would be attributed to the fact that one is more depressed than the other. The Poor Salt test, it's receiving a lot of criticism and an overt attempt to ban it. And this is based on an immobility which develops in animals when they're confined within a tank of water. And antidepressant treatments do prolong the latency to immobility, and I'll describe that in a minute. But the problems with this test as a model of depression is that depression is a multifactorial chronic relapsing disorder, whereas these rats will be perfectly all right once they're dried off and go back to their home cage. Also, antidepressants reduce immobility after subacute treatment, unlike the therapeutic lag in humans, which takes weeks or months. There's also a dispute over whether the behavioral despair, as this immobility is called, is actually a depression-like behavior. If so, what aspect of depression, or whether it's an element of passive coping, that survival time increases if you don't swim. And what really pulls the carpet from under the feet of the model is that some groups in the planet use it as a model of anxiety, not depression. So where does this idea that behavioral despair is depression-like behavior come from? Well, actually, it comes from the groups who developed it in the 70s. And these are excerpts from 3 of their papers. We suggested that the state of despair is resembling depression. Another one, it may reflect a state of lowered mood. Finally, it was hypothesized that immobility reflected animals having learned escape was impossible, given up hope, and it was therefore given the name behavioral despair. So there never was any evidence that the forced swim test induces depression, and that remains the case. But the forced swim test is an excellent screen for antidepressant drugs. As you can see, all the catecholamine targeting drugs prolong immobility, prolong the latency to immobility in this test. And here we have just one example of a paper each for rat and mice. Even the newer antidepressants show up as a positive result in this test, including ketamine, which, as you know, is an anesthetic agent. In a sub-anesthetic dose, it prolongs the latency to immobility. You wouldn't expect that of an anesthetic agent unless this test really has predictive validity. So how can you have a test where antidepressants show a positive result if it isn't a model of depression? And this is what Garner would call the logic trap, which goes like this, that if all antidepressants make animals sing the national anthem, then some people are assuming that then when animals are not singing the national anthem, they must be depressed. And that's what is happening with these models. So how can you have a screen for an antidepressant drug that isn't a model of depression? And that's illustrated on the left-hand side. This is a bridge in Warrington. Poor old Warrington, it's flooded again today. But several years ago, the bridge was washed away. Call that depression. And the parents on this side of the river couldn't get across to the other side to get their children to school. If that's depression, then the solution, antidepressant, was to get in the car, drive up the river to another bridge, down the other side of the river, and deliver their children at school. So it's perfectly possible for depression and antidepressant to have different neuronal networks and follow different routes in the brain. And that would reflect how the brain works, as in sleep versus arousal, appetite versus satiation, and pain versus antinociception, for instance. So it's quite possible for the forced swim test to score antidepressant without it being a model of depression. And if that's the case, then we can't assume that understanding how antidepressants work will tell us anything about the causes of depression, because they involve different neuronal networks. So in summary, there are two reasons to use an animal model in preclinical research. One is to study the causes and process of the illness. Now that assumes that the animal has the illness and the abnormal behavior is analogous to the human disorder. And that needs cautious interrogation and validation through translation and back translation. They can also be used as predictive drug screens for new treatments. The abnormal behavior does not have to relate to any aspect of the disorder of interest, but the validity requires continued consolidation or qualification for false positives and false negatives through translation into humans as each new drug class emerges. So in summary, an experimental procedure can produce a behavioral model that's valid for one purpose, but not another. And that's what we're going to discuss today. So thank you. And I'm now going to hand you over to David.
Hello, I'm David Heal. And in this part of the presentation, I'm going to be talking about the translational validity of animal models in safety pharmacology assessments. We heard from Clare that animal models are extensively used in mode of action and efficacy studies, but they also have an extremely important role to play in tolerability and safety testing. One of the differences is that mode of action and efficacy are generally validated by clinical trial data. However, in the case of tolerability and safety, there is generally no clinical trial validation, and as a consequence, translational validity is extremely important because the data from both the non-clinical studies in efficacy and tolerability and safety play an important part in the drug approval process. They are essential for a realistic benefit risk analysis, and they also will help to define the risks to patients, the public, and society. This is what happens when we get it wrong. These are the annual US overdose deaths in the years from 1999 to 2017. And you can see that there are pretty shocking statistics here, with up to 30,000 people dying of opiate overdose deaths in 2017. What's very important when we are thinking about translational validity is many of these medicines are prescription pharmaceuticals, and therefore understanding the risks that they pose to patients and the public is absolutely essential. These are some key animal models for investigating psychological safety profiles. These are part of the CNS safety pharmacology testing portfolio, and all these tests must be conducted to good laboratory practice standards. They include the Irwin profile, which looks at the behavioral and neurological adverse events of drug candidates; drug discrimination, which looks at the similarity of the psychoactive effects of the drug candidate relative to those of known drugs of abuse; intravenous self-administration, which looks at the potential to induce psychological dependence; and withdrawal-induced physical dependence, which is self-explanatory. The information from all these non-clinical safety pharmacology tests are important when defining drug labeling. Their information can appear as part of the warnings and precautions in the product label. And in addition, if there is a significant risk of drug abuse, the drug may be scheduled as a controlled drug and its prescribing restricted. In Europe, the European Medicines Agency issued a guidance for the non-clinical investigation of the dependence potential of medicinal products in March of 2006. All of the tests which I presented on the previous slide are mentioned in this guidance. I'm going to focus on one test in particular to illustrate the points about translational validity. This is the intravenous self-administration test. In this test, rats are placed in 2 lever operant chambers, and they are initially trained to lever press for food rewards, and one lever is paired with the food rewards. Once the animals are competent in this task, they are surgically implanted with an intravenous catheter in a jugular or femoral vein, and this is then linked to an infusion pump. When the animal is placed in the chamber, when it presses the lever a fixed number of times, this results in the delivery of a small quantity of the drug in the syringe. If the animal finds that the effects of this drug are rewarding, then the animal can continue to lever press to receive further infusions. This shows the translational validity of the intravenous self-administration test. A whole raft of drugs from various classes of substance of abuse are shown in the far left-hand column. This covers all of the major classes of known drugs of abuse, and examples are shown in the column adjacent to it. In the next 3 columns, you can see the potential for human abuse and whether the drug will serve as a positive reinforcer in the intravenous self-administration test, in rats and in primates. All of the text which is shown in black indicates there is congruence between the risk for human abuse and whether the drug will serve as a positive reinforcer. You can see that there are only a few exceptions, and these revolve around the cannabinoids and the hallucinogens. So this test has extremely high predictive validity. That being said, it can generate some false positive results. In this table, we can see that there are false positives, and these include the dopamine agonists and also the dopamine reuptake inhibitors. And these are drugs which serve as reinforcers in both rats and primates, but are not subject to human abuse. There is a good physiological reason for this. The false positives are probably created because the rats have no emetic reflex and the one in monkeys is extremely weak. The animals therefore get the rewarding part of the pharmacology of these drugs without their propensity to produce nausea and vomiting. In summary, therefore, animal models have an important role not only in identifying new drug targets for drug discovery and as predictors of therapeutic efficacy, but also as indicators of drug safety and tolerability. In this part of drug development, translational validity is critical because in safety pharmacology testing, the data from the clinical trials will not serve as a safety net if serious drug-induced adverse events or the potential for abuse and dependence are missed. From a risk mitigation standpoint, it is better to use a behavioral model which is too sensitive, i.e. it generates some false positives, than one which lacks sensitivity, i.e. generates some false negatives, because it puts patients and the public at risk. In the workshop, we will be exploring some of these aspects of translational validity. Thank you for your attention.